Welcome to Faith in Science. I'm Dr. John Ashton. One of the things, of course, we can notice on people are their ears. And recently I was with a group of people, and it was interesting how I just noticed that the shape of ears can be quite different. But when you look at the shape of the ear and all the contours and little grooves, this is in the outer ear, the fleshy part of the ear. It has this particular shape about it. And so while all ears on different people are sort of different, some of the outer shapes, the overall inner construction, and there's a general pattern to the shape of the ear. And of course that applies to all our different organs and our noses and eyes and this sort of thing. They have this basic functional shape, and which is related to their function. And recently I read a very interesting article on the ear by a scientist that has written some chapters wrote a chapter in my book In 6 Days, Professor Andy McIntosh, and he's a mathematician with both a PhD doctorate and a DSc, Doctor of Science doctorate. Very clever mathematician, and he's very interested in design in nature, and he co-authored a book a few years ago called The Wonders of Creation, Design in a Fallen World, and it was co-authored by Dr. Stuart Burgess, and Dr. Stuart Burgess is another professor at a university in the UK, in England, and also they were co-authored with another person, Brian Edwards, looking at all different aspects. And one of those aspects they looked at was the amazing design in the ear, the incredible design in the human ear. I know I've spoken about this in the past, but reading this article refreshed and pointed out some interesting things that I certainly wasn't aware of. And of course, the Bible talks about the hearing ear and the seeing eye. The Lord has made them both. That's Proverbs 20:12. And so we know that the ear actually, like the eye, is powerful evidence for intelligent design. It must have been designed. Now, we know how sound or any noise that we hear is the result of little compressions in the air. So, and these compressions in the air, there'll be a high pressure followed by a low pressure, and depending on the depth of the pressure, or the increase in pressure and the decrease in pressure, these and the rate at which these little increases and decreases in pressure occur, which is called the frequency, the number of these pressure cycles per second is the frequency, what we call the frequency. And depending on the frequency is the type of sound that we hear. And so what happens in our ear, of course, is we have this amazing detection system to pick up and detect these sounds. Now, some of these sounds are produced by very, very small changes in pressure, extremely small changes in pressure. And as a result then, that those changes in pressure have to be amplified up and detected, and then that change in pressure has to be converted into an electrical impulse. And of course the device that does that is scientifically called a transducer. So the ear mechanism is an amazing transducer that converts mechanical pressure energy into electrical energy and retains this signal that then goes through to the brain so that we hear things. And when we look at the ear, we know, the ear, we have this outer part of the human ear, the decorative part, and then that leads to this canal, this little opening that goes into our head, which is, of course, the ear canal. And at the end of that ear canal is the eardrum. And that's why it's very important for, young children not to stick, something in their ear, one of mum's knitting needles or something like that, because they can puncture that eardrum and that can lead to, deafness. And along the lining of the ear canal, there are little ligaments, and then just behind the eardrum is a little hammer system, and 3 little bones called ossicles, and then part of those is another little bone called an incus, and then right behind that is another little bone called a stirrup, the stirrup. Now these bones, the little malleus or the little hammer, and then there's a series of little lever bones. It's a bit hard to explain this, of course, without a diagram in front, but if you can imagine you've got the eardrum, and then connected to that eardrum are a series of tiny little bones, the smallest bones in the human body, that are connected in the back of that to that eardrum, so that any little deflection in that eardrum is actually magnified through a series of levers and through to the stirrup. And, of course, that part where those little lever system is called the middle ear. And, of course, the middle ear has a drain system. The Eustachian tube comes away from that. Now, the little stirrup system then is part of another amazing amplifying system that connects to the cochlea, which is a long tube, a coiled up tube that extends around, and at the end of that tube, of course, we have the organ of Corti, and it's quite a fascinating structure. When look, I mean, even within that itself, there are all sorts of membranes and so forth within the cochlea. If we unwound the cochlea and all the membranes and bases that are and parts that make up that particular tube are quite amazing. And so those vibrations that the eardrum picks up are transferred, as said, from they hit the eardrum, they're transferred through the tiny little bones into the cochlea, and then the complexity of the parts that make up these components are actually all there's inner parts within, like within the lever system. We talk about these little bones, but those little bones have little joints, little levers, and they're special cells that are made up that allow the movement at these times without falling apart and so forth, the hinging, all these sort of mechanisms. And what astounds me is that all these structures are encoded for in the DNA. So the DNA code, which is made up of 4 types of chemical structures, which are again arrangements of atoms that make up a particular chemical, which we call a base. And there's 4 of these bases that we give the names A, C, T, and G, the abbreviations of 2. These stand for the chemical names. And using combinations of those chemicals all strung together, those combinations can be read by an amazing molecular machine called a ribosome, which is made up of over 300,000 atoms, of different types of atoms. And this machine can read the sequence of those letters and then arrange proteins and other compounds together to help build the cells that make up this structure, that make these bones, and they grow in place in just the right place. And one of the fascinating things is that these little bones, for example, that we talked about there, that make up the inner ear, those little amplifying bones, little lever systems, a little hammer and so forth, they don't increase in size after we're born. So up until we're born, they're growing, and then when we're born, they don't increase in size anymore. I think it's quite fascinating. And so this ear in humans, the ear canal is about 20 millimetres long, not quite an inch long, 4/5 of an inch long. In other animals, of course, a different size and can be longer. And as we talked about earlier, the human ear can hear these different frequencies. So these little pressure pulses, the human ear can hear from about 20 little pulses per second up to nearly 20,000 little pulses per second. But as we get older, we tend to lose that ability to hear those very high pulses. I know I have hearing tests reasonably regularly as part of work, health and safety, and I know I don't hear much above about 16,000 cycles per second. And so I've lost that ability to hear those very high sounds. But of course, young people can hear those very high sounds. On the other hand, if we look at the ear system in dogs, they can hear not quite as low as us, as 20 little pulses per second, but from about 65 pulses, but they can hear all the way up to 44,000 pulses per second. So that's amazing. Actually, just reading the article here too, with cats, and I don't know how many animals they've tested, but apparently cats can hear from 55 pulses per second up to 77,000 pulses per second. So that would be, ultrasound. We couldn't hear those pulses, but animals can hear those pulses. It's quite interesting that the human voice can produce these beautiful range of harmonies, typically between about 2,000 pulses per second to 5,000 pulses per second. One of the interesting things that I read about was how we hear water running. And it's interesting, it appears that research that's been done reasonably recently, actually, was only published in 2015 on the proceedings of the meetings on acoustics conference that was heard. And this was a study looking at ocean bubble acoustics and looking at, a sort of when you use sound to clean things. And it appears that the sounds of running water is a result of tiny little bubbles in the water popping, tiny little bubbles of air. And as these little bubbles air, they vibrate at a frequency that depends on their size. So flowing water produces a range of audible frequencies so that we hear the sound of a bubbling brook or a flowing waterfall or crashing waves. And these are made by these air bubbles that are released at that time and that are vibrating, and they, of course, then vibrate our ear, and that's how we hear the sounds of these waters. I thought it's really, really amazing. One of the things is that our hearing range is appropriate for us to hear these sounds of nature. It's quite fascinating. I think that our hearing range is appropriate for us to enjoy music, to enjoy speech, and so forth. It's interesting how the cochlea, if reading how it's sort of like this seashell type coil that's coiled up, and inside that cochlea is sort of a membrane, and of course this membrane tapers along the cochlea. So it's a bit like a xylophone. If you look at a xylophone, notice that the little bars on the xylophone that you tap with the hammer to make the notes tend to be different lengths. All the way along they're different lengths, and that's how they produce the different sounds. So it's the same with inside the cochlea there. We've got these membranes that this tapers, and this allows the different parts of the membrane actually will pick up, and they'll have what we call a natural frequency of vibration, and so they will resonate and in that way amplify. So different objects have what we call a natural frequency of vibration, and that's why you may have seen a picture of a bridge in the United States that during wind it began swinging, and as it was swinging, the rate at which it was swinging, the natural vibration of the swinging, happened to coincide or was very close to what we call the natural frequency of the structure. And what that means is that it absorbed the energy, and so each vibration added to each vibration. It's sort of like when you're pushing someone on a swing. If you push them a little bit, and then you just match when they came, you push them again just a little bit, their swing will keep on increasing, and eventually they're swinging really big. But unless you push them just at the time when they've just about stopped, and you push them again, if you push them halfway up the swing, it's not going to amplify them. You'll interfere with it. And so if there's a natural frequency, and this sound is coming in at a natural frequency, by having the membrane at tapering, that means parts of the membrane have these different natural frequencies, so it absorbs the energy and therefore amplifies it. So it's quite amazing. But then, of course, we have the device at the end that converts this vibration into electrical engineering. And this is one of the areas that really challenges and fascinates engineers. I remember when I was working at the BHP research laboratories, we were looking at some of these transducers and converting mechanical energy into electrical energy, particularly when you're looking at very, very fine amounts of electrical energy. And so what happens is we've got the cilia there, and these cilia, these tiny little fine hairs. Now, these hairs that compose the cilia there are absolutely amazingly fine. The thickness is in the order of about 1/70th of the thickness of a fine human hair. So some human hair is quite coarse. So if you get a fairly fine human hair, these little cilia, these tiny little hairs that are part of that nerve at the end of the cochlea there, they or in the cochlea, they vibrate and they operate. It's quite interesting. A little membrane that touches the cilia, and this actually opens a tiny little trapdoor, push a little trapdoor attached to a spring. And this spring, its tiny little spring-type structure, is only a few nanometres thick. Now, a nanometre is a millionth of a millimetre. And so this is a tiny little spring only a few millionths of a millimetre thick, and it's about 100 millionths of a millimetre long. And one end pulls on this tiny little trapdoor, which is on the side of one of these little hairs. And this is one of the smallest examples of springs in the natural world. Now, what this does is this tiny little spring, this tiny little mechanical spring, right, which is operated by the mechanical energy that is coming from the sound vibration. So the sound is a pulse of air. Sometimes when you're in a really, really loud concert, you actually feel physically those pulses of air. So, but when there's a tiny little, very, very faint sound, like a little moth flying past your ear or something like that, then that is amplified and operates one of these little springs, and it opens this little trapdoor that allows charged ions, and ions are charged atoms that are flowing in a fluid called in this fluid-filled cochlea, and the movement of those ions excite little nerves. And so the ions are carrying electric charge, and they excite the little nerves, and that sends a signal then, an electrical signal. So the charge on those ions affects then, or generates a tiny electrical current, passes along the nerve to the cerebral cortex in the brain. And it's interesting, there's nerves, there's about one nerve for a whole range of hertzes. So in the upper range, there's about 2 to 3 different cycles per second per nerve ending, up around your 16,000 cycles per second, this sort of thing there. The one nerve would cover, say, 16,001, 16,002, 16,003 vibrations per second, whereas down in the lower end frequencies, like 60 or 100 cycles per second, there's a single nerve for each different cycle per second. I find this absolutely fascinating, this structure. You know, we look at ears and, and some people have their ears pierced and all this sort of thing. But behind that outside structure, as we go into this structure, there's an amazing structure there. Now, evolutionists have to believe that this amazing structure arose by chance somehow with all these different parts. But when you think about it, unless all the parts there, they're not going to work. And the codes are so complex, like the structures for the little lever systems. And again, we can draw the little lever systems, draw schematic diagrams of them and so forth. We don't realise that there are different cells involved in all the different, as I mentioned earlier, the little different joints and all these sort of things, all the different components that make up those different structures. All right to talk about the cilia and how fine they are, but all the cells that make up these cilia all make the little tiny little springs, right? And we can be amazed that, they're so tiny. But let's hang on. We've got a code that has to make the molecules that make up the structures, the long-chain polymers that coil in such a way and have that springiness in that particular situation. And then we have to have the cells that make up the little hinge on the little trapdoors. And then we have to have the code to make the fluid that makes and have the ions there, the charged atoms there at just the right levels. There's codes to make all these different components that are there. And these codes are using the letters A, C, T and G. They look nothing like those structures, but combinations of those codes are then interpreted by a code-reading machine that enables the cell to build all those parts. And as those first couple of cells come together, as the first male and female gamete cells join together, whether it's in a dog or a cat or a human, and they grow, those cells are programmed at the right time, some of those cells will begin to form the ear of that animal. Now, evolutionists have to believe that that amazing mechanism arose by chance mutations. And we know it's absolutely impossible. We don't see new types of sense organs evolving in any animals or anything. Everything stays the same. It's powerful, powerful evidence for a creator. But there's more. I was reading an article on bats, and I learned something about bats because, bats have amazing ears too. And it's interesting, you've got the big bats. And I used to learn, bats use sonar and emit sounds and so forth to detect their prey. Well, one of the things I learned was that the big bats actually don't do that. I didn't realise that. There's only very few species and only one genus use echolocation. That's in the big bats, what they call the megabats. However, the microbats all use echolocation. And one of the interesting things about echolocation is when you think about it, and they use sound ranges beyond that of the human ear that humans can't hear. But it's interesting that bats emit these sounds, but it's extremely intense, so intense that when they emit these sounds, and they have different ways, some emit them through their tongue and different sounds, different types of fleshy appendages and so forth, and their nostrils to make these sounds are so intense it would send them deaf. But it's very interesting. The bat's ears are designed in such a way that the middle ear muscles contract when the bat is calling so it doesn't deafen itself. And then the muscles relax so the bat can hear the resulting echo, which is, of course, of much lower intensity because it's being reflected off something. And I thought, isn't that amazing that there's so much design in nature that makes it work? Otherwise, the bats would have gone deaf. And yet evolution has to assume that these things arose by chance. It's I think there's so much evidence there. So we have so much evidence in nature for God creating us. And, as the Bible just clearly states, it's in Proverbs, the hearing ear and the seeing eye, the Lord has made them both. Proverbs 20:12. How true. Evolution can't explain the origin of those things. You've been listening to Faith and Science, and remember, if you'd like to re-listen to this program or any of the past programs, just Google 3abnaustralia.org.au and click on the listen button. A number of different programs will come up there. I'm Dr. John Ashton. Have a great day.
You've been listening to a production of 3ABN Australia Radio.